Yes, Parvati, you can share
Yes,
your
ma'am.
PPT. Yes, Parvati.
Okay, ma'am. Ma'am, ma'am.
It was visible. What happened?
Now,
No,
ma'am.
no, now it is not visible. You have to share once again.
Okay, ma'am. Ma'am, it's visible, ma'am.
Yes, yes, it is visible.
Good morning, all. My name is Parvati. I'm presenting the presentation stomatic hybridization and its utilization crop improvements. Next is introduction. Stomatic hybridization was first introduced by Carlos and Ital in Necatona glassica. Stomatic hybridization is an important tool of plants breeding and crop improvement through the production of interspecific and intergenic hybrids. Techniques involves in the fusion of protoplast of two different genomes followed by the selection desired stomatic hybrid cells and generation of hybrid plants. Protoplast of sexually sterile haploid, triploid and aneuploidy plants can be fused the produced fertile diploid and polyploidies. Next is what is stomatic hybridization? It is development of hybrid plants through the fusion of stomatic protoplast of two different plant species or varieties is called stomatic hybridization. Next is protoplast. Protoplast term is proposed by Einstein in 1880 to refer to the entire cell and including the cell wall. Next main points related to the stomatic hybridization is involve the stomatic cell and tissue culture is essential, equal contribution of cytoplasm over the barrier of cross incompatibility. Next is novel combination of genes from unrelated species. Next, types of stomatic hybridization based on taxonomical relationship. Interspecific hybrids. Interspecific hybrids means hybrids between two different species of the same genus. Example, Datura. Intergenic hybrids. Intergenic hybrids means hybrids between the plants of two different genera of the same family. Example, potato plus tomato. Interterrestrial hybrids. Next, techniques of stomatic hybridization. These are the techniques of stomatic hybridization. First is isolation of protoplast. Next, fusion of the protoplast of desired species or varieties. Next, identification and selection of stomatic hybrid cells. Next, culture of the hybrid cell. Last is regeneration of hybrid plants. Methods and materials for isolation fusion of protoplast. Successfully stomatic hybridization was generally obtained through the PEG. PEG means polyethylene glycol. Fusion more recently electrofusion techniques have become available. First direct step method. Next first we take a leaf segment and incubate that overnight 15 to 18 hours with enzymes mixture of 20 degrees Celsius and freeze gently liberated protoplast. It mixture into a filter through the screw gauges and remove the desired transfer debris. Transfer into a 13 into 1000 millimeter screw capped tubes and centrifuge at 100 gram for 1 minute. Next, protoplast form a pellet and supernated was removed. Next is the process is repeated for 3 times and form a protoplast will obtain and washed with the 13% sorbitol solution with later replaced into a sucrose solution for 20% and centrifuged at 200 gram for 1 minute. Then clean is the protoplast which will flow on the in centrifuge. Next, we taken out into a bulk. Next, advantages of stomatic hybridization: production of novel interspecific and intergenetic hybrids, that is product promata, hybrid of potato and tomato. Next, production of fertili developed hand polyploidy from sexually sterile haploid, triploid, and aneuploidy. Transferred a gene into her diseases resistance and stress resistance and many other quality characteristics studied on the lead of plasma gene. Next, limitation of somatic hybridization, poor generation of hybrid plants, non-viability of used products, not successful in all plants, production of unvariable hybrids, lack of an efficient method for a selection of hybrids, no confirmation of expression of particular triads in somatic hybrids. Next, conclusion. The somatic hybridization can be accepted too much quieter than the genetic engineer because traditionally breeding must be inverted between the biotechnology events, that is protoplast fusion, and the release of product for the market. Valuable resistance to various pathogen or environmental tissues stresses can be identified into a direct somatic hybrids and may have been entered into a current plant breeding programs. This is the conclusion of this experiment. Thank you, ma'am.
Okay, Parvati, one thing I wanted to tell you, you can, yeah, stop sharing. You can
Okay,
stop
ma'am.
sharing. Yeah. What you have explained or whatever you have done in your slide is only you have explained what is somatic hybridization. What we wanted you to do it was that you have to take any one work of any one person who has whichever one sample, say one plant they have taken and they have done somatic hybridization. So you are supposed to do with the papers, research paper. And what you did was, what is somatic hybridization? What are the different types of somatic significance of it? This is like you have explained what somatic hybridization is. But we wanted something else from you. I think all of you know this is not the first time you're doing it. Umpteen number of times we have told you it is the research papers. Research papers. Please underline that. It is with the research papers you are supposed to do it. Anyway, next, who is there for the next PBZ?
I'll present it, ma'am. Vijay Lakshmi.
Vijayalakshmi, PBZ, right?
Yes, ma'am. Yes, ma'am.
1 minute. Okay, you can share.
Yeah, ma'am. Screen is visible, ma'am.
Yes, yes.
Good morning, everyone. My name is Vijay Lakshmi from PBZ. Today I am presenting about enhancing the fuel properties of beverage waste as non-edible feedstock for a biofuel production. This was written by Andy Shaw and Linoil JR News, received on 20th Jan 2021, accepted on 25th April 2021, published online on 2nd June the same year. Abstract. A few industries such as beverage industries have experienced some growth in sales during COVID-19 pandemic. Globally, beverage companies generate over 200 million tons of biomass annually, which largely ends up as animal feed or in the landfills. With the UK government commitment to reduce its carbon footprint by at least 68% in 2030. This paper unlocks the potential for improving the fuel properties of beverage waste through a blending process and explores the determination of optimal fractions for the blends via characterization with an initial moisture content of 82% and 58%. Brewery spent grain and spent coffee grounds pre-treatment reduce the moisture. The study concludes the biomass blending improve the properties of biomass. Introduction. Oil prices witnessed significant stagnation during the current year for two main reasons. One is oil price war between the two of the world's strongest oil producing countries, and the second one is COVID-19 pandemic. The world lockdown 2020 in March, government issued a stay order at home. For instance, the loss of the soil industries is more worsened. Some industries such as beverage and personal protection equipment industries experienced substantial growth in 2020. The rise in beverage sale led to the increase in biomass waste. In likewise, coffee shops and bars, restaurants were closed, which caused a fall in sales of coffee. Coffee being the second and the brewery grains being the fifth in the trade commodity. Processing these products generate large amount of waste. Recovering these waste for energy use, enhancing their fuel properties and reducing the amount of landfills. Rich biomass waste resource from the beverage industries and to channel these resources for the energy use. Major drawback is high moisture content. It requires densifying these biomass type. The resulting of product in an inexpensive lingulossic biomass with an enhanced fuel properties. Materials and methodology. Brewery sample. The brewery samples were provided in the form of shaft, and the coffee waste was provided in the form of particular powder. The samples were collected and stored immediately in the freezer box to prevent microbial growth. Methodology. First one is proximate analysis of biomass material, in which proximate analysis is a widely acceptable method for biofuel characterization. It is cost effective, it prevents the moisture and a fixed carbon, volatile matter, and ash content of the biomass. Moisture content. The moisture content was determined using a British standard BSEN 147743 as 2019. They prepared sample in dried at the temperature of 105 plus or minus 2 degrees Celsius in an air atmosphere until the constant mass is achieved. The percentage moisture is calculated from the loss in mass of sample. Ash content. The ash content present in the biomass material was determined using BSEN 14775/2 2009 by calculating the mass of the deposit after the sample was heated in a furnace at temperature of 550 plus or minus 10 degrees Celsius in the air under controlled condition to 1 hour and cool down at room temperature. Volatile matter content. Volatiles and fixed carbon waste determined according to the same in 2000 according to same BSEN 15148 2009 standard. The volatile matter evaporated during the heat. Difference in weight represent the volatile matter which is present in the biomass waste. Fixed carbon percentage. This is the mass of the residue left after the release of volatile matter. Ultimate elemental analysis. The elemental analysis was carried out using a Perkin-Elmer 2004 elemental analyzer to measure the different elements This study applied the carbon, hydrogen, nitrogen, and a sulfur mode, which permits the calculation of these four elements in the organic matter, that is in a biomass, while the amount of oxygen was determined by subtracting the sum of the other element present from 100%. And a calorific value analysis. A calorimetry procedure was used to evaluate the higher eating value of the biomass material. Phenolic content analysis. The phenolic compounds were measured using the method detection limit. The method detection limit is the minimum detection of the limit of the substance that can be measured and reported with 99% confidence that analyte concentration is greater than zero, and it is determined from the analysis of the sample in a given matrix containing that analyte of the biomass. And densification of biomass composite into a pellets or a bricklets. The decision to utilize the biomass for energy application depends on the energy profile and the moisture content, calorific value, and density. The use of actual application of certain types of biomass dependent on these variables. Torfraction process for biomass composite products. Torfraction is the thermochemical process involving the interaction between the drying and incomplete pyrolysis result. That is, the decision of use of biomass for energy use depends on the energy profile, moisture content, and the calorific value. These characterization involve the proximate element and the ultimate and the calorimetric analysis. The matrix of these sample has been found to interfere with the result of this test. The sample was therefore diluted by 20x to improve the signal to noise ratio, and is in doing so the detection of limit for the test was also evaluated. By conclusion, the result shows that biomass blends improves the fuel properties, and the blending process could be identified as from form of pretreatment to enhance the fuel properties. Wet torfraction, as already demonstrated of high potential to convert a wide range of biomass, with emphasis on biomass with a higher moisture content to energy dense solid biofuel. In this, the biomass waste can be used as a biofuels for the future generation to increase the carbon footprint in the atmosphere. These are the some of the references. Thank you, ma'am.
Excellent. Vijayalakshmi, I'm very, very happy. You have taken such a good topic, and this is what is called as SSR report.
Thank
So,
you, ma'am.
yes, very good. Very, very right way of doing it. This is what we ask a student to pick up a research work, and you tell how they have worked on, so that this will help you to do your work in future when you are doing any research. Excellent.
Thank you, ma'am.
Yeah. Okay, next CBBT. Who's doing it?
Ma'am, Pooja, ma'am.
Okay, Pooja. You can share your slide.
1 minute, ma'am.
Yeah. Pooja, you have to share, ma.
Ma'am, I'm sharing it from the Google Slides, ma'am. 1 minute.
Okay.
Ma'am, it is not visible there.
No, we can see, but it is not coming as sharing.
1 minute. Then I'll join and share. Now, ma'am.
Yes. You can show your slide. Yes, we can see.
Good morning, everyone. I am going to present on the topic in vitro regeneration of Bacopa moneri, a highly valuable visible medicinal plant. Coming to the introduction, India has a great diversity of medicinal plants, and these medicinal plants are used in Ayurvedic, Unani, and a number of pharmaceutical products. And Bacopa moneri is one of them. It belongs. It is commonly called as Brahmi or Neer Brahmi. And it contains many components like alkaloids, saponins, sterols, and phytosterols in it. Bacopa moneri is placed second in a priority list of most important Indian medicinal plants on the basis of its medicinal properties, commercial value, and potential for further research and development. It is important to conserve this medicinal plant through different techniques, and the rapidity of multiplication of true to type plants and efficient transplantation of Bacopa moneri can be useful in conservation and propagation of the allied plants. And it has a vast and varied applications. Coming to the materials used in this research paper was plants of Bacopa moneri, twin 20, a surfactant, 0.01% of mercury chloride, MS medium, and a plant growth regulator called BAP. Methodology. The source of the plants were used as explants for the in vitro regeneration. Selection of nodal, internodal, shoot tip, and leaves were taken as explants. And these were then treated with running tap water followed by surfactant twin 20 and washed with mercury chloride and repeatedly washed with sterilized water. Then it was cultured on MS medium with different plant growth regulators at pH 6.8, supplemented with nutrients, iron, vitamins, agar, and agar as a gelling agent. The explants were inoculated among the explants. Only nodal explant had responded, and the nodal portion was inoculated and supplemented with different concentration of BAP. For shoot initiation, they were supplemented with BAP and NAA, and the regenerated shoots were subcultured every 3 weeks in the same media composition. For rooting, 3 to 4 micro shoots were of 2 to 3 centimeter were cultured on in vitro rooting medium, supplemented with IBA alone and recorded every week. Coming to the result. After 2 to 3 weeks of culture, explants showed growth response in different culture medium and different parts were used as explants material, but fast and good response was observed in nodal explants. The initiated explants were implanted on shoot multiplication medium containing BAP with combination of NAA showed multiple shoots within 2 weeks of inoculation. By the protocol described here, we can obtain 5 to 6 rooted plants from one explant in 90 days and it increases the rapidity of multiplication of true to type plants and efficient transplantation of this medicinal plant, Bacopa moneri, can be useful in conservation and propagation of the allied plants. The figure A and B shows the shoot initiation on MS medium, C and D shows the shoot multiplication, and E and F shows the root formation in MS solidified medium. These are the references. Thank you.
Yes, good, Pooja. See, you people, when you start looking into, these kind of researches, you will get a better idea as to where you can look into your, and in future where you can start working. So it should be your, focus should be from now itself towards the research, especially those students who are really keen on, want to work on and go for your higher studies. So please, girls, start thinking on that line also, so that it will help you in future, and always look for the latest work. See, that is very important. What is recent research work which is happening? That is very important. Good, Pooja, very well presented.
Thank you.
Yeah. So, who is the next from PBZ?
I will present, ma'am.
Okay, Monisha. Yes, you can do it.
Ma'am, can you see my slide?
Yes, yes.
Good morning, everyone. Today, my topic is vegetative propagation of allspice, and this is the article published by J. Rima, P.A. Matthew, and B. Krishnamurthy, Indian Institute of Spices Research from Calicut. Abstract. Allspice is a hardwood species and it is indigenous to West Indies. And for this, different methods of vegetative propagations, namely cuttings, air layering, grafting, and stooling were attempted to study the effectiveness of various auxins, and influence of season on routing has been studied. And a combination of auxins like IBA and NAA at 2500 ppm prepared with charcoal was also effective for the routing of the all species. And coming to introduction, all species, that is Pimenta dicola. It is also known as Jamaican pepper, which is indigenous to West Indies. And the allspice flavor, it resembles the mixture of cinnamon, clove, nutmeg, and black pepper. And it is a hardwood species, and it is valued for its dried unripeness. But the mature berries, these berries are used for the spice purpose. And the main objective of the study was to standardize the successful clonal propagation technique in allspice through all the four techniques, that is vegetative propagation of all four techniques. And to see the effect of auxins and influence of season on routing of cutting and air layering has been studied in this article. And the materials and methods were used are, they have used four different methods, that are cutting, air layering, grafting, and stooling. To observe the routing in any one of this method in allspice, there were used total, they have used six treatments, namely IBA 1000 ppm and IBA 2000 ppm, NAA 1000 ppm, and NAA 2500 ppm. um, all the hormonal the samples were collected for all the four experimental methods, and the hormonal formulation were prepared in with activated charcoal for all the methods. From all the above auxins, the hormone required for the plant, the respective hormonal treatment, that is NAA 2500 ppm and IBA. NAA is nothing but naphthalene acetic acid, and IBA is indole butyric acid. For the treatment was given for each month, and each method and activated charcoal, they have used the activated charcoal without any disturbance. And they provided all the treatment for a particular method which was required. And the treatments were repeated at two intervals for 1 year. After 1 year, the observation and routing were made for each methods, for each method, air layering, cutting, stooling in allspice. They studied after 1 year. And they studied the effect of season and also the influence of hormone, that is auxin. And in results and discussion, they have seen routing in allspice, 64 to 56% was observed during the month of February and December. By the combination of, they have seen NAA at 2500 ppm. This was very effective. And highest routing was observed during December to February, and the routing was lowest in the month of August. And in air layering, they have observed that October was the best month, which has provided 100% success of routing, and maximum routing was observed during October to December, and least routing was observed during April to June. And in grafting, the mature and juvenile cycles of allspice on its rootstock was successful with 56 to 80%. And successful graft showed that 100% establishments in pot. And stooling, this is also successfully practiced in a related genus, namely Sidum juana was and also it was
Cydium guajava.
effectively yes ma'am,
Cydium.
Sidum
You know what is Cydium
guajava.
guajava?
uh, no ma'am.
Guava. Guava.
Okay ma'am.
Fruit. Guava fruit, no? Sepekai.
Yes ma'am, yes ma'am.
Ah,
And
yes.
in this stooling method was also successful in allspice. And conclusion, they have concluded that they started they in conclusion they have concluded that all the vegetative propagations like cuttings, air layering and grafting and stooling, it was successful to maximum of 64.4% to 85%, and all the methods were successful for this allspice species. And these are the references. And thank you.
Very good. Very good, Manisha.
Thank you, ma'am.
Okay, who's the next?
Ma'am, I'll do it, ma'am.
Okay, Navya. Very good.
Is my screen visible?
Yes, yes.
Okay. Good morning, ma'am. Good morning, everyone. My topic for today is the effects of nano TiO2 on seed germination, development, and mitosis of root tip cells of Vicia narbinosis and Zea mays. This research article was taken from the Journal of Nanoparticle Research and written by Monica Ruffini Castiglione, Lucia Giorgetti, Chiara Jerry, and Roberto Creminoni. This study aimed to provide new information about phytotoxicology of nano-TiO2 on plant systems to contribute to the evaluation of potential harmful effects of the nanoparticles on monocots and dicots. They considered their effects on seed germination and root elongation by applying a concentration range from 0.2 to 4% in the plants of two species. The aim of this study was to test the effects of TiO2 is nothing but titanium dioxide nanoparticles, to evaluate their effects on two plant species, which is Zea mays L, which is a monocot. It is nothing but your corn crop, and Vicia narbinosis L, which is a dicot, which is commonly known as narbin beans. Nano-sized titanium dioxide is widely used in cosmetic and skincare products, mainly in sunscreens or sunblocks and toothpastes, and in antibacterial and cleaning air products, and for decomposing organic matter in wastewater, since its properties of high stability, anti-corrosion, and photocatalyst activity. So, materials and methods. So, the seeds of the two species were washed overnight in tap water. They were then soaked in distilled water and then in a titanium dioxide nanoparticle suspension at four different concentrations, which were 0.2, 1, 2, and 4% for 24 hours. Then all the samples were transferred into petri dishes and they were let to germinate until 72 hours in controlled environmental conditions. 60 seeds for each treatment of each species were utilized for germination tests and for root elongation capacity. Moreover, after 3 days of treatment, 5 roots for each treatment were fixed in ethanol and glacial acetic acid in the ratio of 3:1 for mitotic activity analysis. At least 1,000 nuclei were randomly selected for each slide, and they observed these under a microscope. So coming to the results were that nano TiO2 treatments, it was significantly affecting the germination rate, but only in the first 24 hours of imbibition. No germination process was detectable in Vicia narbinosis following nanoparticle titanium dioxide treatments, while more than 20% of controlled seeds were germinated within 24 hours, whereas in Zea maize, the seeds reached 50% germination at 0.2 concentration, but at higher concentration, the germination rate was almost halved. So by this, the discussion says that after short-term exposure and under experimental conditions, it is seen that titanium dioxide affects seed germination in both the species, delaying the process during the first 24 hours in Zea and blocking it completely in Vicia. But afterwards, both the plant systems started to recover their germination ability uh, at 42 to 72 hour window, and but they are not able to grow with the same vigor as the controls of higher concentration. So these are the references. Thank you.
Good. Good, Navya.
Thank you, ma'am.
Next, from CBBT.
Pragna.
Okay.
Ma'am, is the screen visible?
Yes, screen is visible. Your PPT is not visible.
Yes, ma'am.
Yes, now it's visible.
Good morning, one and all present here. I've chosen a paper on the topic anther-culture. Anther-culture is a technique by which the developing anthers at a precise and critical stage are excised aseptically from unopened flower bud and are cultured on a nutrient medium. The microspores within the cultured anther develop into callus tissue or embryos that give rise to haploid plants. And this technique involves growth of the anther, disturbing its natural habitat and environment of the enclosed anther. The title of the paper that I have chosen is The Effect of Incubation Period and Culture Medium on Pepper Anther Culture. The pepper meaning the bell pepper, the scientific name of pepper is the Capsicum annum. Um, this paper was written by Stanislava Grozeva and Anmol N. Nankar. It was accepted in the year 2019. Um, introduction The increasing demand of multipurpose peppers have prominently encouraged pepper breeding community to study diverse aspects of pepper genetics, physiology, breeding, and biotic stress. This uh, current study was aimed to assess the in vitro androgenic response of two prominent Bulgarian varieties, that is the um, one is the Strayama variety and the other is the Latin Medal 7 variety in 5 different duration times, 12, that is 12, 14, 16, 18 and 20 days on incubation medium and regeneration mediums containing kinetin in different concentrations and combination of zeatin and indole acetic acid. And uh, coming to the experiment, the two varieties of peppers, that is the Strayama and the Latin Medal 7, were selected and were grown in the glasshouse condition for almost 7 months. And the flower buds providing anthers with microspores from late uninucleate stage were sterilized in 5% calcium hypochlorite for 20 minutes and rinsed thrice with sterile distilled water. Next, the anthers that were excised from the flower were incubated on induction culture medium, that is the Murashige and scoop medium. The petri dishes with the anthers uh, were treated in darkness at 35 plus or minus 1 degree Celsius for 12 days without any addition of the growth regulators, and this was named as the regeneration medium and it was considered as a control. The effect of incubation period on the induction medium and different regeneration medium variants were analyzed for improvement of embryo induction and plantlet regeneration. The first experiment was combined with 3 concentrations of kinetin in regeneration medium and 5 different duration times on incubation medium. The uh, regeneration medium that was considered as control was named as 12, and the the next medium, which is the culture medium variant 12-1, it contained zero point 0.1 mg per liter kinetin, and 12-2 with 0.2 mg per liter kinetin, and 12-3 with 0.3 mg liter per kinetin. And the second experiment was combined by regeneration medium supplemented with 0.25 mg per liter zeatin and zero point 0.1 mg per liter IAA, that is the indole 3 acetic acid, and this was named as variant 12.4, and another 12.5 variant with 0.5 mg per liter zeatin and zero point 0.1 mg per liter IAA, and these were incubated for 12, 14, 16 and 18, 16, 18 and 20 days, and the results were analyzed. The results from this study clearly demonstrated positive influence of prolonged anther incubation period and induction medium on embryo formation and plantlets regeneration. The uh, highest frequency of embryogenesis was observed from Striama at 16 days cultivation, whereas after 20 days treatment uh, for Latin Medal 7 variety had the highest frequency of embryogenesis, and the medium variant containing low concentration of K and combination of zeatin and IAA had the highest regeneration frequency. These are the references of the papers. Thank you, ma'am.
Very good. Thank you, Pradnya. Good presentation. All of you are doing very well. Who's the next person? Next, who is doing it?
Ma'am, I'm doing it, ma'am.
Sneha. Okay. Yes, Sneha, you can share.
Yes, ma'am. Ma'am, is my screen visible, ma'am?
Screen is visible. Yes. Yes.
Good morning, all. I am Sneha from third year PBZ, and today my topic is on floral development in bolting garlic. So the authors are Rina Kemensky and Hem D. Rabinovich. Abstract. Gallium, which is the Latin word Elem sativum, is a completely sterile plant propagated only vegetatively. The main aim of this research was to study the sequence of morphological processes occurring during floral initiation and development of a number of bolting garlic accessions from the allium gene bank, which is taken in Israel by using SEM, scanning electron microscope. The garlic inflorescence is an umbel-like flower arrangement, and the branches, which are flower clusters, of which arise from a common meristem. The number of flowers have a distinct morphological typical of the genus Allium. Flower stalk elongation precedes the swelling of the apical meristem and its subdivision into several centers of floral development. Within the clusters, floral primordia develop unevenly. So differentiation of topsets begins after floral differentiation on the peripheral part of the apical surface, and the size, number, and rate of development vary among genotypes. At least 4 morphological types differing in flower or topset ratio was distinguished among the 12 clones, which is studied in this investigation. So, introduction. Uh, Garlic, which is a completely sterile plant and is therefore propagated only vegetatively. So, increase in genetic variation occurs only through random or induced mutations or by somaclonal variation, and new cultivars are bred by clonal selection. Sterility was assumed to research from degeneration of the tapetum, degenerative like diseases induced by organisms such as rickettsia, mycoplasma, or viruses, chromosomal deletions, or competition for in nutrients between floral and vegetative buds in the development inflorescence. Indeed, in bolting of garlic, fertility restoration was achieved by continuous removal of developing topsets, assigned garlic and some other alien species. Uh, the alien species are Cerulum, Allium cerulum, Allium proliferum, Allium venial, Allium carinatum, Allium scorodrasum. To this group of viviparous plants in which topsets develop instead of plants or intermingle with flowers in the inflorescence. The sexual sterility of garlic inhibits or markedly reduces its improvement for economically important traits, including resistance to pests, higher yields, and better quality. Hence, the restoration of fertility to garlic has been attempted by many researchers, and it has been suggested that the presence of vegetative toxins is the major cause of sterility. So, bolting in garlic occurs in plants exposed to the appropriate environmental conditions, and the clones are classified according to their ability to bolt. Uh, so, there are three types of bolting. One is complete bolting, in which the plants are long, thick flower stalk and with many flowers and top sets. In complete bolting, the plants produce a thin, short flower stalk bearing a few large top sets and usually forming no flowers. Non-bolting, the plants are normally forming a flower stalk but producing only cloves instead of incomplete scape. The effect of environmental conditions on inflorescence formation in garlic has been partially described for several commercial garlic clones. However, little is known about the morphological events during apex differentiation and during floral initiation and differentiation in garlic. It is generally accepted that the inflorescence of alien species appears as a terminal umbel. The floral development of Allium mole, subgenus Allium, and Allium odorum, subgenus is Rhiziridium, has been described as branching of floral meristem. Light and scanning electron microscopy of floral development in Allium rothii, Allium altissimum, and Allium caratavenus. Subgenus is Melanochromum have revealed that apical meristem differentiates into several peripheral swellings, so thus producing flower clusters. In other species of Allium, the individual garlic flower consists of perianth lobes, which are 6 stamens and tricarpellary pistils situated in the innermost whorl. Methods and materials. The method used here is the materials used are plant material. The bulbs of 12 clonally propagated bolting garlic accessions were received from the field gene bank for vegetatively propagated in a short day alien species, which was conducted in Israel, Rehovot. The general pattern of fluorogenesis is described for the clone 2091, which was introduced from Russia. In addition, inflorescence structure of garlic, Allium sativum 2030, 2031, 2045, 2096, and 2229 was introduced from Russia and Kazakhstan, and 2216 introduced from China, as well as Allium longicapsus 3001, 3002, 3003, 3004, and 2011 was introduced from Central Asia, which was studied prior to spathe opening. And storage
Shreya,
and
can
growth
you go to
conditions.
the result slide, ma? Go to the result slide.
Okay, ma.
Observation, yeah.
So observations formed here are so it consists of the leaf formation. In garlic, leaf primordia begin as protuberance as a apical dome. In each leaf primordium develops into a partly lifted, complete ring. The tubular sheets of older leaves envelop the successive younger leaves in pattern similar to foliage developing in the bulb of onion and shallot. So this is the images of in which leaf formation is found, and this is a figure shows the floral initiation in garlic with spathe removed, and image B, it's the differentiation of the centers of development in a reproductive meristem with spathe removed. C is the floral scape in which 10 mm long of leaflets are shown, and D is the inflorescence becomes hemispherical in shape and consists of numerous floral primordia. And the second observation is florogenesis. After the formation of 6 or 7 leaves and leaf primordia, the subject is exposure to inductive environmental conditions in which apical meristem of garlic becomes reproductive. During this transition, the spathe develops quickly and envelops the apical meristem just before floral initiation. So, in many allium species, inflorescence differentiation occurs prior to scape elongation. So, however, initial elongation of the flower stalk precedes the swelling of the floral meristem, and the differentiation of floral initiations begins only after the scape reaches 5 to 7 mm in length, and the apex diameter exceeds 0.5 mm. Later, the apical meristem subdivides to form several swellings, each of which gives rise to a number of individual flower primordia. And this is the images in which scanning of electron microscope graph of garlic floral development is seen, in which florogenesis is also seen. And the image A shows the differentiation of floral primordia is uneven. Floral parts occur in oldest floral primordia. And the image B shows floral primordia in the outer wall of flower parts arises. Image C shows the floral pedicels and leaf-like bracts elongate. And the image D shows the newly developed meristems, which is appeared in the base of the inflorescence. And the third observation is the genotypic variation.
Yeah, you can just move to the next slide. Actually, you should have concised it, Sneha. You have made it very, very big and elaborate. It should have been concised. Yeah, go to the conclusion part, results and conclusion or discussion. What is the discussion? How do you conclude it? Sneha, can you hear me? Girls, can you hear me?
These are the images in which image primordia about in the early stage of differentiation, and the image. Hello?
Yes, yes.
Can I change it now? So finally, the conclusion is that the garlic inflorescence is considered as a bulb like flower, and the branches arise from a common meristem in which the numerous flowers have a distinct morphological typical and of the genus Allium. The 5 whorls of floral organs each having 3 members, and in many Allium species the garlic inflorescence develops from the apical meristem, and under the present experimental conditions, florogenesis commenced after the development of 6 and 7 leaves and initiation of the spathe.
Okay. Presentation is good, Sneha, but then you should reduce the content. Anytime, whenever you are giving these kind of presentations, you have to read it, understand, and put it in bullet forms. That you
Okay.
so that has taken more time for you, but next time make sure you're doing like that. Okay.
Yes, yes,
Yeah.
ma'am. Thank
Okay,
you.
good.
These are the references, ma'am.
Okay.
Thank you, ma'am.
Okay, thank you. You can stop sharing. Anybody else who wants to do it within 5 minutes time, then we can do it. Otherwise, we'll conclude for today.
Ma'am, I'll do it, ma'am.
Okay, Yashoda. You have any class now?
No, ma'am.
Okay, then I think we can have this and then we'll conclude. Yes, Yashoda.
Ma'am, is my screen visible, ma'am?
Yes, yes, it's visible.
Okay. Good morning, all. Myself Yashoda from PBZ. Today I'm going to present my self-study report. So the article which I have chosen is the effect of polyploidy and hybridization on the evolution of floral color in Nicotiana. So this article is by Elizabeth, Arnold, Laura, and Baldwin. This is from Department of Botany and Plant Science, University of California. The article is received in the year 2014 and it is accepted in the year 2015 and it is published in the 2015. So I'm going to explain the synopsis of the article. The article goes like this. The main aim of the article is to investigate whether the change in floral color can be accompanied polyploidy and homoploid hybridization. Homoploid hybridization is nothing but the hybridization without change in the chromosomal number. So, which is an important process of angiosperm's evolution. So that for this article, the team members they have taken the corolla tissue of Nicotiana, was examined by spectral reflectance. The spectral reflectance is nothing but it gives the results of the presence or absence as well as the position and shape of the specific observation feature, and even based on the spectral shape which is corresponding to the pigmentation in order to access the pattern of floral color in the evolution. So polyploidy and homoploid hybridization spectrals were compared with those of their progenitors. Progenitors is nothing but ancestors or the parents to evaluate whether hybridization has result in the floral color shift. So the floral colors categories in Nicotiana seem to have arisen in multiple times independently during the evolution of genes, and most younger polyploids are displayed on unexpected floral colors. The floral colors is weakly constrained by phylogeny in evolution, and the color shift due to occurs in associated with the polyploid and homoploid hybridization. So the article will conclude that the evolution of floral color has been driven by or resulted by the pollinator shift. The variety of pollinators which are visited the plant Nicotiana is moth, birds, bees, and bats. This is the graph which shows the roughly corresponds to color perceived by human observation through spectral reflectance. These are different floral colors which is seen in the plant Nicotiana. Then these are the references. Thank you, ma'am.
What was the result? Just conclude on that. What was the result? Go back. Go back to the slide. You
Ma'am, the
didn't.
conclusion is, the evolution of floral color is because of the pollinators, ma'am.
Pollinators. Okay. Fine. Okay.
Ma'am,
Yes.
Ma'am, actually, we don't have a class now, ma'am, after this.
Is it? But I have to. Okay, anybody else wants to do it? Maybe another 10 minutes I can spend.
Ma'am, do we have a class in the afternoon?
Yes, you will have.
Okay, ma'am.
Anybody else who wants to do it?
Ma'am, can I do it?
Yes, Prakriti, you can. Prakriti,
Yes, ma'am.
uh, 1 minute. Okay, you start.
Okay. Ma'am, can you see my screen?
No, you need to share.
Ma'am, can you see now?
Yeah.
Good morning, all. So, my article topic is qualitative
You
analysis of
haven't shared
wine.
your presentation.
Prakriti, presentation you should open.
Can you see now?
Yes.
Good morning, all. My article topic is qualitative analysis of wine prepared from banana and orange. Introduction. So, various fruits have been used for the production of wine since the dawn of human civilization. The fermentation with yeast is used for the production of wine, that is considered as one of the oldest alcoholic beverages. Usually, grapes were taken as the substrate for the winemaking, but in recent years, preferences have been given to other fruits such as apricot, banana, citrus fruits, and so on. Banana and orange are highly perishable in nature. The fermentation is a low energy prevention
Can you
process.
change the slide? You have to move the slide, I think.
No, ma'am, here only.
Okay. No, because
Increase.
it's showing the title. I think you should be moving your slide.
Can you see now?
I can see your first slide only. I mean the second slide.
Introduction.
No, not introduction. Others can see the introduction?
No, ma'am.
Ah, yes.
So I'll keep like this only, ma'am, then.
Yeah, you keep it like keep scrolling the slides. That's
Okay,
fine.
ma'am. So this study focuses on the preparation of wine from banana and orange along with the physiochemical and organoleptic evaluation. Materials and methods. Collection of materials, ripened bananas and oranges, and wine yeast.
Preparation of pre-fermenter culture. Diluted solution of banana juice extracted from the pulp was taken as a banana must. This must was treated with 0.05% of amylase and 0.05% of pectinase for 24 hours and filtered and pasteurized. 0.05% of wine yeast was added. It was incubated at normal room temperature for 4 days. This was the pre-fermenter culture for banana wine. Similar process was carried out for orange pre-fermenter culture, except there was no dilution and the must was treated with 0.05 pectinase only. Preparation of wine. The juice extracted from a dozen of bananas were diluted by the addition of two-third amount of distilled water. The sugar concentration was maintained to 20% Brix by the addition of sugar as per Piaget's square law. It was treated with amylase and pectinase for 24 hours, then filtered and sterilized by the addition of 50 ppm potassium metasulfate. To this 5% banana pre-fermenter culture was added, which was then incubated at room temperature for about 3 weeks. The main process carried out for the preparation of orange wine was similar to the banana wine. However, the must was not diluted and it was treated with pectinase only. Determination of physiochemical characteristics of wine. In the incubation period, its TSS, pH and titratable acidity were determined on each consecutive day. The alcohol content was determined only at the end of the fermentation. Study of organoleptic characteristics of wine: color, smell, taste, clarity, texture, and astringency of the wine was studied. The sensory quality of banana, orange, and standard wine was examined by members of the selected panel, in which 6 sensory assessors participated in the wine tasting panel. Results. The change in the value of total soluble solid during the fermentation of banana and orange wine is shown in the figure 1. The initial TSS of banana was 16, which dropped down to 10 within a week. It further kept on decreasing until it was 6 at the end of fermentation. In the case of the orange, the initial TSS was 10, which dropped down to 8 on the ninth day and remained constant till the end of fermentation. The value of pH during the fermentation of banana and orange wine is shown in figure 2. The initial pH of banana was 5, which dropped down to 4 till the end of fermentation. In the case of orange, the initial pH was 3, which remained constant till the end of fermentation. Figure 3 shows the change in the value of titratable acidity during the fermentation of banana and orange wine. The initial titratable acidity of the banana was 0.23. The maximum titratable acidity, 0.4, was observed on the 11th day. It decreased to 0.4 at the end of fermentation. In the case of orange, the initial titratable acidity was 0.41. The maximum titratable acidity, 0.83, was observed on the ninth day, that decreased to 0.32 at the end of fermentation. The organoleptic characteristics were determined using 5 points of hedonic scaling. The orange and banana wine were compared along with the standard wine. The average of individual points given to each wine was calculated and evaluated. It was found that 6 tasters preferred banana wine over orange wine. The results are shown in the figure 4. Conclusion. Hence, the banana and orange both were taken as a substrate with different properties and nutritional value, which resulted in the production of different concentrations of alcohol along with TSS, pH and titratable acidity, although same yeast culture was added. From the result obtained through this study, there was no significant difference in the wine prepared from banana and orange. Also, both of the fruits are readily available throughout the season with a high production rate in Nepal, so banana and orange wineries can flourish in the country. Thank you.
Okay, good. So you can also try this. This is a very, very simple experiment which you people can do it even at home to try. Anybody is interested can try the wine preparation. Okay, good, Prakriti. I think we will end for today.
Thank you, ma'am.
Yes, girls?
Okay, ma'am.
Yeah. So, good preparation. I think all of you have worked hard on this. Somewhere, I think Sneha could have, reduced, or she should have concised the study. She has gone through it, and your PPTs should be very, very precise, and your points, only points should be there. And you should be able to explain whatever you have learned or read through this, the work. So with this, we will end for today's class, and we will resume it in the afternoon because teachers will have other classes. So let's see if the teachers are willing to have it before that. We can have it before. Otherwise, we will have it at 3 o'clock. Okay? No, Sneha, it's okay. It's all a learning process. You don't have to say sorry. It is all learning process. You are all very initial. That is why when you are doing now, when you make mistakes, it can be rectified. No problem. Okay, girls? So I think you can leave now. Thank you all. And others, be
Thank you,
prepared.
ma'am.
We will try and finish with the presentations today. Okay, during the
Yes,
practical
ma'am.
hours. How many more have to do? From CBBT, how many more are remaining?
CBBT around 9 to 10, ma'am.
Okay. And from PBZ?
Ma'am, some people were absent, ma'am, so I don't know about them.
No, if
But
they're absent, see, if they're absent, it is considered 0. Today we will try and wind up because we have practicals. I think we will do it independent from the combinations. So if they are not doing it, it will be considered 0. We can't be, waiting for them to submit it. Okay?
okay, ma'am.
Yeah. So you can share it, tell them that if they're not doing it today, then it will be considered 0. Okay?
Okay, ma'am.
Yeah. So I think we will end the class for today. Thank you all. You can leave, girls, for now.
Thank you, ma'am.
Thank you.
Thank you.
Thank you, ma'am.
Thank you, ma'am.
Thank you.
Thank you, ma'am.